Yeah. Hey, so for this learning module, we're going to be talking about the 7-transmembrane GPCR and the heterotrimeric G protein that it activates, and we're going to go through this signal cascade with protein kinase C being its target. And we're going to do that by explaining the steps and amplification steps throughout this whole process. So let's begin. So essentially, what you're going to start off with is a 7-transmembrane GPCR. GPCR stands for G protein coupled receptor because it is a G protein coupled receptor and will actually be associated on the plasma membrane. Now, since it's a transmembrane protein, this is going to be a key role in activating that G protein on the stalk side. But how the signal cascade is going to start is that you're going to have a ligand which will go to an associated 7-transmembrane GPCR and bind to that receptor. Once this binding occurs, you're actually going to have a conformational change throughout the whole protein. Now, this is interesting because it not only will it change the conformation of the protein on the receptor or the plasma membrane side, but more importantly, the systolic side of the transmembrane protein. Because of this active or conformational change, it will bump into a heterotrimeric G protein drawn here and change its conformation. Now, a heterotrimeric G protein is actually comprised of 3 subunits: the G protein's alpha subunit and the G protein beta and gamma subunits. In this signal cascade, I drew the G protein alpha subunit as itself and the G protein beta and gamma subunits associated together. So, the G protein subalpha unit is actually going to be your GTPase protein, and this is going to be the protein associated with your GTP protein. And so during its inactivated state, the heterotrimeric G protein will be together, but the G alpha subunit will actually have GDP associated on there. Since it has GDP associated on there, it will be stuck in this form. But since we already have this conformational change, it will bump into this heterotrimeric G protein, and it will actually influence the exchange from GDP for systolic free-flowing GTP found inside that cell. So GTP will come in and exchange it for DP, and because of this, the 7-transmembrane GPCR will actually play a key role as a GEF, as you learned in class, a guanine nucleotide exchange factor. Since the ligand binds on this 7-transmembrane GPCR, it's going to actually influence the exchange of GTP for the alpha subunit. Because of this exchange, you'll actually activate the heterotrimeric G protein, and the G subalpha unit and G sub beta and gamma units will actually separate. And we'll go into detail about this on the next slide. But before I do that, I just want to mention that during this whole signal cascade, you will learn about the amplification and how amplification is actually created throughout the cell signal process, and how that in certain steps or key steps on this signal cascade, you're going to have more and more of the effect through small little points like this one right here that I'm going to go over, which is actually that since the ligand is binded on the 7-transmembrane GPCR, it's actually going to activate it, which we already went through the conformational change. But not only does it activate one heterotrimeric G protein, but it can activate many others. As long as that ligand is still associated on that receptor, you could still have many other heterotrimeric G protein still on the systolic side of the plasma membrane and become activated, have its GDP switched out with GTP, and thus separate. I don't know if I mentioned this, but I'll mention it again if I didn't. The G proteins are actually associated on the systolic side of the plasma membrane through lipid anchors. So whenever they move, it's going to be lateral diffusion only. They're not going to pop out anywhere and go like to the nucleus or somewhere. They're just going to stay on that systolic plasma membrane because of lipid anchors. I'm just going to draw a line here. So let's move on to the next slide. All right, cool. So like I said, the G beta and gamma subunits separate from the G alpha subunits, and we're going to talk about the excuse me. We're going to talk about the targets for these two proteins. Now, you want to remember that when they do separate, because the G alpha subunit has GGP now, when they separate, they actually expose their binding sites for their targets. So that's why they don't actually activate other targets when in the inactivated form, because G protein alpha subunit actually has its GDP, and they're still in one subunit together. So the G beta gamma subunit will have actually two targets: an ion channel, which you've probably already learned in class, and we're going to use an example of sodium. So it'll activate the ion channel, and sodium, which is kept on the extracellular side of the cell, high concentrations as compared to the inside, will flow down its electrochemical gradient and just go through that ion channel. The G protein alpha subunit will actually activate a protein called phospholipase C. What phospholipase C is through the name, will actually cut a target which we'll talk about in this module is PIP2. You have to remember that PIP2 is kept on very low concentrations on the systolic side of the plasma membrane. This probably has to do with signaling, and so PIP2 is comprised of two fatty acid domains and a polar head group, which is a 6-carbon ring with 3 phosphates. So phospholipase C will come in and actually cleave that point where the distinction between the polar head groups and the fatty acid chains, and they're going to separate into 2 molecules. And interestingly enough, I'm going to include that there's another amplification process here where the G protein sub-beta and gamma units could actually not only activate an ion channel here, but actually activate a phospholipase C or another phospholipase C. So essentially, you could actually have 2 phospholipase C going in and activating 2 PIP2s. However, we need to remember that PIP2 is kept at low concentrations on the systolic side of the plasma membrane. But it's still really important to understand that there could be 2 targets for this signal cascade, that being an ion channel and PIP2. What I'm going to go over is the ligand that's still here could also be recognized through the amplification step, because since that ligand is still associated with that receptor, you're going to have that conformational change, and you're going to still have that G proteins coming in and being activated by this GPCR. So, interestingly enough, the amplification process has to do with the ligand's half-life properties or half-life capabilities. If it's going to be a long half-life, then you should expect that multiple GPCRs or multiple heterotromic G proteins will probably be activated through the process. So let's move on. So I'm going to let my partner Yona go in with this conversation.
Okay, so once we've activated PLC with the G protein alpha subunit, it cleaves 2 parts of the PIP2, the fatty acid domain and the polar head domain, and these turn into 2 compounds, one being DAG and one being IP3. DAG, being diacylglycerol, stays associated with the plasma membrane because, like I just said, there was 2 fatty acid chains associated with the PIP2, and now it has become DAG. As far as the IP3, once that's cleaved and that the polar head group is now free to roam around, it goes through the cytosol side of the cell, and it actually binds to the IP3 gated calcium channel, which is located on the endoplasmic reticulum. So once that happens, this calcium gated channel undergoes a conformational change and opens up, allowing calcium, which is very high inside of the endoplasmic reticulum, to leak out into the cytosol side, allowing it to increase the concentration in the cytosol side. So once that happens, calcium can either bind to protein kinase C or it can actually go and bind to calmodulin. In our case, we're going to talk about protein kinase C, but as a side note, I would like to talk about how it can go from binding to calmodulin, which then, once bounded to calmodulin, can activate a CAM kinase. This is just another signal pathway like I wanted to mention before we move on to the actual pathway we're talking about in this video.
Okay, so I'm just drawing calmodulin and CAM kinase. I understand that they're not, the best penmanship out there, but like Jonas said, it's going to activate calmodulin, which will actually activate CAM kinase and phosphorylate other target proteins of CAM kinase. So we're going to also talk about how protein kinase C, which is really interesting, what it does, it has two binding sites, one for calcium and one for diacylglycerol. But protein kinase C is actually associated not on the plasma membrane until calcium binds to protein kinase C. Then it will start to move to the plasma membrane and find diacylglycerol and bind to it. So that's an interesting property to note. And what Yona actually left out, which I'll include, is that calcium is actually will be a second messenger that you will learn in class, and although we're not creating a second messenger in this particular step, we're having the availability of the second messenger be produced inside the systolic site, right, Yona?
Yeah.
So
thank you, Mike.
No, you're welcome. So there's going to be high amounts of calcium flowing through that endoplasmic reticulum, which we'll consider as secondary messenger. And so let's go do the last part of this journey. Okay, let me just add a quick layer. And so, you want to take things off?
So, like Mike just previously said, once calcium binds to PKC, now it actually moves up to the plasma membrane side to allow diacylglycerol to also bind to it. It's because PKC has two binding sites to it, and in order for it to be activated, it needs calcium and diacylglycerol to both bind to it and to activate PKC.
That's right. You cannot activate fully the functions of protein kinase C with only just one of its properties, either that being diacylglycerol or calcium 2+. And so only when you have two is when you're going to actually see the properties of protein kinase C and target its target proteins and phosphorylate them. So that's really interesting to note, because if you only have, let's say, calcium and you activate protein kinase C, it goes to the plasma membrane, and you don't have diacylglycerol, you actually will skip out on the effect, and protein kinase C will not phosphorylate target enzyme. And so that is the signal cascade. Oh, what I forgot to mention is, if it wasn't obvious enough, when calcium 2+ goes out of the cell, you're actually having another amplification step here, because you actually have a higher presence of calcium, and then calcium will actually bind to other protein kinases, or, as we mentioned, calmodulin, which could activate calmodulin kinase.
CAM kinase.
CAM kinase. And so that's really interesting to note. However, one interesting little property that I found interesting in class was that, do you guys remember how we had PIP2? Well, when that gets cleaved off, you actually cleave it into only one diacylglycerol and one IP3. So because of protein kinase C's double substrate function, you won't really have an amplification step at this particular step, because you still only have one diacylglycerol, and PIP2, as we mentioned, is kept in very low concentrations on the systolic side. So that's going to be a huge barrier that you want to keep in mind, because although protein kinase C can be activated, it's only going to be a one diacylglycerol for one PIP2 that's cleaved. And so protein kinase C will target proteins, or I'm sorry, phosphorylate target proteins, and you're going to see that throughout the cell signaling cascade. I think that's about it. One last thing we're going to go over is how to turn off this signal cascade, and my friend Yona is actually pretty enthusiastic about this. So.
Oh, thank you.
Oh, you're welcome.
Okay, so like Mike mentioned before, there is a half-life associated with the ligand. So that's one out of the three ways to regulate cell signaling, which is one way to turn it off. Professor Nugent will teach the other two to you guys, but for right now we're going to just talk about the half-life of this ligand. So once this half-life passes for this ligand, and once it pops off this GPCR, now this GPCR is now inactivated. Once that happens, the G protein alpha subunit is actually acting like a GTPase mentioned before in the previous slides, because once that happens, once the ligand pops off, once the GPCR is now inactivated, the G protein alpha subunit changes GTP now to GDP. It self-hydrolyzes itself because it is a GTPase.
That's right. And so once you hydrolyze that GTP, the G protein beta gamma subunit will actually come back, and as well as the G protein alpha subunit with the GDP now, and also come back and form that heterotrimeric G protein we started in the beginning of the module. And thus you could actually activate the signal cascade again. And one last interesting property to note is the endoplasmic reticulum is actually filled with calcium 2+ pumps. And this in part will bring that calcium back down in the cell, used for later signaling, so calcium will just, over time, be pumped back in the endoplasmic reticulum. And so, that should be it for this learning module.
Thank you, guys.
Thank you.
Have a good day.
Good luck with your finals. Good luck.